wherever you are. So thank you for joining today's H3ABioNet-Fogarty Collaborative Research Seminar. So my name is Rolanda, and I will be hosting today's webinar. Just a few housekeeping rules before we start. We have two speakers for today, and the questions will be taken at the end of both of the talks. You are most welcome to type your question in the chat box, and I will read them out. Please be sure to state who the question is directed at, but you are also welcome to ask your question using your audio. Please just raise your electronic hand to alert us before you do. And right now I will ask Verena to just make Martha a co-host, that she could start sharing her screen. Martha will be our first speaker for today. And while they are doing that, I can introduce Martha. So Martha is a MSc student at Pwani University in Kenya, but she's based at the Kemri Wellcome Trust Research Program, where she undertakes her research project. So Verena, can Martha share her screen?
Martha, you can go ahead and share your screen now.
There you go. So over to you, Martha. You may begin your presentation.
So hello everyone, my name is Martha Luka, and I'll be talking to you about my master's research project, which is elucidating human rhinovirus transmission pathways at different scales of observation. So the reason that we need to study human rhinovirus, or HRV, HRV is one of the most common respiratory viruses within the human population. It's associated with a broad spectrum of disease, mainly the common cold. It's also linked to at least a third of asthma development and exacerbations, and scientific evidence has more recently linked it to the lower respiratory tract infections, which are more severe. Genetically, it's a positive sense single-stranded RNA virus classified within the family Picornaviridae and the genus Enteroviruses. It's also a very diverse virus. We currently have 169 genotypes in circulation globally, and these genotypes are antigenically different. So we have 169 serotypes within species A, B, and C. It's a relatively small virus with its genome being 7.2 kilobases, and it's a single open reading frame. So the top image here on your right is just a snapshot of its capsid structure. It's about 30 nanometers in diameter. And this is its genome organization. So I'd like to bring your attention to 3 regions of interest. We have the 5' untranslated region. And this is actually the target region for molecular detection assays. This is because we have a segment of it that is conserved across the genotypes. The other part is the other 2 regions are the VP42 junction and the VP1. These 2 have both been used for assigning the virus to their respective genotypes. The VP42 is made up of the entire VP4 region and a part of the VP2, and it's actually the most sequenced region. It's more sequenced than the VP1. So now to the background of my study, which is to understand transmission dynamics across different scales. I've mentioned that we have 169 serotypes in circulation. So it becomes very difficult, or this diversity frustrates vaccine development efforts, because we need to have a vaccine that can actually give serological protection against all these serotypes, and that at the moment the efforts are not promising. So our infection management strategies now are shifted to controlling transmission. However, human communities are complex in the sense that we have different population structures which are characterized by different contact patterns. So in this cartoon illustration, I just illustrate the 4 population structures of interest to this study. We have the households, schools, primary healthcare facilities, and referral hospitals. within households, we see a certain type of contact. So for example, we have an age-structured contact whereby children within households or between neighboring households tend to play together, for example, after school or even over the school holidays, and this shapes how infections are transmitted within the community. We also see a cross-generational type of contact patterns, for example, children with their grandparents or even their caregivers, who in this case will be adults. So households are particularly important in trying to understand what is going on in the larger community. School-age children, on the other hand, have actually been shown to have the highest contact rates within the community. And studies have proposed them to be the most likely introducers of infections within the households. So this study is also going to try and see if we get the same result or a different result. Primary healthcare facilities have been shown to be pretty efficient ways to monitor what is going on in the community, particularly for respiratory and diarrheal outbreaks. So primary healthcare facilities are a relatively efficient way to pick up such outbreaks within the community. And on the other hand, referral hospitals tend to handle the more severe cases, most of the times referred from the primary healthcare facilities. So these are the 4 settings that my study aims to look at. And just continuing on the background, I've mentioned why we need to study transmission and how our communities are complex. So I just need to point out that the studies that have looked at the molecular epidemiology or transmission of rhinovirus, most of them just pick one population structure and try to study it. But so we have inadequate information in linking one social structure to the other social structure. And so this leaves us with sketches on what is going on in the community as a whole. And also these studies tend to use the VP42 sequences I mentioned earlier, which are a relatively short region of the genome. The VP42 sequences tend to be about 400 bases long, and sometimes they are inadequate to resolve transmission. So I want to bring your attention to the phylogenetic tree on the right. This was a household study done by Kamau et al. And they identified numerous genotypes circulating within households. And these are demonstrated. So this is a species A, rhinovirus species A tree only. So if I bring your attention to S75, which is the most frequent genotype, you see that all sequences are within a straight line. This is just to indicate that we do not have nucleotide substitutions across all these sequences. And so we cannot differentiate infections from one individual to the other individual, and therefore cannot make any inferences on transmission. So this raised the question as to whether the region, the VP42 region, is too short to make such inferences, which is why we aim to compare what we see on the VP4 and the whole genomes. And my study aims to inform computational models which will predict outbreaks and assess the effectiveness of public health measures. So my overall objective was to elucidate human rhinovirus transmission pathways at different scales of observation by sequence data analysis, where the first specific objective was now to use partial sequence data to establish these transmission pathways. And then the second objective was to optimize a lab assay for the whole genome sequencing of HRV using next generation sequencing. And the third was to evaluate the benefit of whole genomes over partial sequences in studying the transmission and evolution at the different scales of observation. So now I want to briefly describe how I went to select my samples. My samples were selected from previous studies that investigated rhinovirus using partial sequences. So we had a study looking into households, another looking at outpatient clinics, another looking at inpatient clinics, and another for school. And my study is trying to compare these inferences from these different studies. And so because of the high diversity of human rhinovirus, it doesn't make a lot of sense to just compare all genotypes to all genotypes, because then it becomes very noisy. So that is why we aimed to pick one genotype per scale and just zoom in and see if we could make any clearer inferences. So for the household study, I picked the most frequent genotype that was identified, which was HRVC35, and it was 44 samples across 3 neighboring households in Matsangoni sublocation, which is in Kilifi County. Then for the outpatient clinics, I selected HRV-A15, which was actually also the most frequent genotype identified by this study. And I added four sequences that had been identified in the Kilifi County Hospital, which is the referral hospital in Kilifi. So these, the HDSS are the primary healthcare facilities, and this is a referral hospital. So this was 66 samples. For the A22, we had 11 referral hospitals at different sites across Kenya. And in this case, we also settled for the most frequent genotype. The approach for HRV-A11 was, however, slightly different. Instead of going for the most frequent genotype identified by the school study, I just need to point out that the school that was investigated is located within the Junju sublocation in Kilifi. So we wanted to pick a genotype that had actually been identified in both the school and the outpatient clinic, and to see how the two settings compared to one another. And this was now the genotype that was identified in both, but just to point out, it was looking at each of these, it wasn't the most frequent in each of the settings. So in total, I had 160 samples, or which had already been sequenced for the VP42. So I may say samples or sequences, just not to confuse you. And these were across 4 scales and 4 genotypes. So the concepts that I implemented in this study, the first one is phylodynamic inferences. This is where we get to use phylogenetic inferences to be able to infer transmission. So if you, and of course, so for example, in this phylogenetic tree, we have, you can be able to tell between these sequences labeled A, B, C, and D, how if we get to see the most closely related sequences, and from that we can be able to tell A infects B or A infects D and such information. However, because even when you try to generate a tree with the most divergent sequences, you will still get some form of clustering. We needed to have some tests of significance to improve our confidence that this is indeed a transmission pathway. And so these tests of significance were bias factors and posterior probabilities, because we mainly use the Bayesian framework. Haplotype networks, on the other hand, are intuitive ways to visualize transmission or just the distribution of alleles within a population. And most of the times they are used alongside phylogenetic trees, not on their own when it comes to transmission inferences. And we also use spatial networks, whereby now we just incorporate the phylogenetic input into geographical maps for a more intuitive visualization of the virus pathways. So now to my first objective, which was now to use the partial sequence data to identify the transmission pathways. I used multiple sequence alignment using MAFT, and then I had a discrete Bayesian phylogeny using BEAST version 1, and the spatial networks were generated using spread3 and the BEAST output. Then I generated haplotype networks using POPAT, and had also calculated the sequence identity using MegaX, and used all this to make inferences. So now I will briefly take you through some of the results that we found. Within the household, household sequences had the highest sequence identity, which was 99.9%. And as you can see from the phylogenetic tree on the left, all sequences clustered by household of origin. So you can see there's a very nice clustering. The haplotype network identified 2 main alleles, that is this circle here and this other. And one allele was entirely limited to household 5, while the other was shared between 51 and 19. And the numbers in brackets just indicate the number of nucleotide substitutions between one allele and the other allele. And using the phylogenetic branching patterns, so that is the distance from the tips to the most common recent ancestor, we were able to identify that children below the age of 12 years were actually the most probable introducers of rhinovirus to the household. So I think it was a 3-year-old, a 7-year-old, and an 11-year-old, all of them under 12 years. These are the most probable introducers of rhinovirus to the households. Then comparing what was going on in the school and the community. So by community, I refer to the Junju Outpatient Clinic, which serves the community where the school was located. And it's actually a reflection of what goes on in the community. So we were able to identify a sequence identity of 96%, that is all the sequences. But of course, if you look at the haplotype network here, you see that the school sequences were more identical compared to what is seen in the larger community. So we see much more diversity in the larger community than in the school. And then we see that there is also a clustering based on the origin of the sample. And the school sequences appear to stem from the community. So instead of having all sequences mixed up, it's like what this suggests, given the high posterior probability here, is that what is in the school is actually just introduced from the community, and afterwards we see a within school transmission pattern. Then looking at the larger Kilifi health and demographic surveillance system. So these are the primary healthcare facilities that are distributed within the Kilifi health and demographic surveillance system. And we were able to see a sort of geographical clustering pattern. So if you see sequences, for example, in Mavueni, in Sokoke, in Gerenya, Masangoni, these are the different sublocations within the KHDSS. However, we also have some sequences randomly interspersed. So for example, if you see Chasimba, seems to be all over the entire tree. So we see both sort of mixing, but also some form of geographical clustering on the phylogenetic tree. And the spatial network, we were able to see that we have significant epidemiological linkages between both neighboring and distant locations. So an example being, for example, Pingilikani here is strongly linked to Mavueni, and we also have areas such as Chasimba being linked to Matsangoni, which is way up north. So we have both transmission that is local and the other that is not very local. Then looking at the country, we had 11 sites across the entire country. So I grouped this into Eastern, Central, and Western to just reduce the components. And we were able to see that there was also a geographical form of clustering, whereby sequences from Western appear to and cluster together, Central, Eastern. Just to point out that we also seem to have a phylogenetic group here that there's some sort of mixing. So similar to the KHDSS, we have both geographical clustering and some sort of random, in quotes, random interspersing across the phylogenetic tree. And in the spatial network, we were able to see that majority of the clustering was within Western Kenya, but we also had distant linkages, for example, Kilifi and Kakamega. And this had the least sequence identity, which was 93%. So to conclude, my first objective is that households have fewer virus introductions compared to larger geographical spaces. This is demonstrated by the high sequence similarity that we see within the household sequences. Then we also see that school-age children are the most probable HRV introducers to the households. And phylogenetic clustering was based on geographical locations suggests local transmission. So now to take you through my second objective, which was to optimize a lab assay for the whole genome sequencing of human rhinovirus using next generation sequencing. The approach was to use an amplicon-based enrichment for the virus. So we generated genotype-specific primers targeting amplicon sizes 1 to 1.6 kilobases long across the entire genome. And so we acquired sequences that were publicly available from GenBank and used the Genius software to generate these sequences, to generate the primers, sorry. So this is the lab assay that we were able to set up. We were able to document or rather describe an end-to-end lab assay that includes RNA extraction. We were able to optimize the monoplex reactions from the initial set of primers. If you look at the gel here, you'll notice that we have a lot of non-specific bands, but this is because at this point we are still trying to get the PCR conditions right. And once we did that, we were able to multiplex all pull reactions of different primers into one reaction. And this is the aim was to generate a cost-effective assay that reduces the number of reactions that we need to run per sample. And then we had a library prep, the Nextera library prep, and we sequenced using the Illumina MiSeq. And we had a genome assembly. So so just to point out now, from this assay, we were able to capture the entire rhinovirus genome. So this image is just showing you that we were able to capture the entire rhinovirus genome, and the read depth varied across the genome randomly, which is probably as a result of the library fragmentation dynamics. Our newly designed method for human rhinovirus whole genome sequencing can successfully enrich for the virus in clinical samples with variant virus titers. The pooling of reactions enabled a more economical assay than singleplex reactions, and we have not yet tested, but we believe that it can be adapted for Oxford nanopore sequencing, which we see an increased uptake of recently due to its portability. And now to my third objective, which was to evaluate the benefit of whole genomes over partial sequencing in studying the virus transmission and evolution at different scales of observation. So here I show maximum likelihood tree using sequences from the Kilifi Health and Demographic Surveillance System. So these are the sequences that were successfully sequenced for the whole genome sequences, just to make sure that I was comparing like with like. And we see that areas that were initially now totally identical, such as this, are now being resolved in the whole genome sequences. And with this now, we can now be able to differentiate infections from one individual to the other individual. So this is a better measure to be able to infer transmission. Further analysis is still ongoing in this comparison of whole genomes versus partial. And we hope to refine these findings. Also, we were able to identify recombination breakpoints in both structural and unstructural regions of the genome. If recombination is a very important aspect to study prior to generating phylogenetic trees, because failure to take it into account can lead to erroneous inferences on phylogenetic trees. So, and the molecular clock as well. So this is a very important aspect to study when studying the evolution of viruses. So we were able to conclude that whole genome sequences provide more information than partial sequences, particularly in evolutionary dynamics such as recombination, and they can better resolve transmission because now we are looking at a higher number of nucleotides compared to the partial sequences. So with that, I would like to acknowledge my supervisors, Professor James Knox, Dr. Charles Agoti, and Dr. Ali Kitty, the Virus Epidemiology and Control Team at the Kenry Wellcome Trust, Ian Bitt and the NIH, IDEAL, and Pond University. Thank you.
Thank you so much, Martha. All right, so I think we are still good on time, so we'll move on to Sounkou. So Sounkou, may you please get your slides ready to share, and I'll introduce Sounkou. Sounkou Touré is a student, an MSc student based in Mali at the University of Science, Techniques and Technology in Bamako, and he's also an affiliate of the African Center of Excellence in Bamako, in bioinformatics of Bamako. Thanks, Sounkou. Over to you.
Can you see the screen?
Yes, we can see your screen.
Okay. Morning, everybody. My name is Sounkou Mohamed Touré. I'm a MS student in bioinformatics at the African Center of Excellence in Bioinformatics, and also a a Fogarty International Traineeship Fellow at Touré University. Today I will be presenting my master's thesis project, which is about the identification of genetic loci for blood lipid levels in individuals of African origin. I will begin by introducing the topic, give the methodology, give some results, and discuss them. As you know, cardiovascular diseases are a class of blood and heart conditions, which include diseases like stroke, heart failure, hypertensive heart disease. It is a third causes of mortality worldwide, with 41% of all deaths according to WHO in 2016. And we note that in the epidemiology, we note a decreasing mortality in high and middle income countries. The epidemiology of the disease in sub-Saharan Africa is that it is the second cause of mortality in Africa after HIV/AIDS, with totalizing 13% of all deaths. We also observe a higher mortality and increasing cases and younger age profile of cases on the African continent. Moreover, we observe a higher incidence of CVD in population of African diaspora, like African Americans. Among the causes of cardiovascular diseases is dyslipidemia, which is an abnormal concentration of blood lipid level in general, and more specifically, which are the molecules which are readily measured are high and low density lipoprotein cholesterol, triglycerides, and total cholesterol. These blood lipid level have been shown by twin and family study to be influenced by both environmental factor and genetic factor. The environmental factors include factors like diet, physical activity, medication, and infection like HIV/AIDS or alcohol consumption. And the genetic in the family study, there are mutations in genes like ApoE, LipG, and CTEP have been shown to explain the segregation of dyslipidemia cases within families. We know that dyslipidemia is a common condition, common disease in Africa in general, and there is a marked higher prevalence of dyslipidemia among people having chronic conditions like HIV/AIDS, hypertension or diabetes, which explain the comorbidity of dyslipidemia with other chronic diseases. Moreover, in African American, we observe that despite African ancestry being associated in genetic study with better blood lipid profile, which does not translate for them to lower incidence of CVD. Several explanations have been put forth to explain in fact, but among them is the fact that there is evidence of genetic interaction involved in this case. For instance, we see that for the effect of triglyceride on triglyceride, the effect of a loci on triglyceride at LPL loci depend on whether or not the person carries a African or a European haplotype in this loci. So, but all the other causes, biological causes of this difference remain unknown. Up to date, few studies have investigated the genetic determinants of common cases of dyslipidemia in Africa. A study in 2018 suggests that about 71 polymorphisms in 28 genes were linked to dyslipidemia, but most of these studies were based in North Africa, 70%, and the polymorphism did not translate in sub-Saharan Africa, and this study design was hypothesis driven because of the cost of typing. A more data-driven approach to discover the biology of complex thread is genome association study, which is an observational strategy to test the association of genetic marker against one or more complex thread like AIDS or diseases like stroke. which are being successfully applied across complex stress and diseases. But in GWAS, we know that there is a neuroscientific bias in the study participant. Usually, with regard to lipid GWAS, we see that African ancestry individuals are being mainly used for either a replication of findings in Europeans or fine mapping. And the African, including the studies, tend to be African Americans and not people from the continent. And the larger GWAS to date of a multi-ethnic cohort of blood lipids reveal thousands of genetic markers associated with the blood lipid level in a classical gene like statin drug target PSK9, apolipoprotein, and apolipoprotein A. But there was another study denoted a lack of transferability of major loci of blood lipid level, particularly for triglycerides in a Uganda cohort. There were heterogeneity of effects which were observed in the multi-ethnic studies of lipid thread. And up to this day, there were only one Pan-African meta-analysis of lipid GWAS. The causes of because there are different causes of this lack of transferability of effects of risk loci, which are among other frequency and linkage disequilibrium differences between population. Because in GWAS, allele which are associated with a threat are not expected to be the causal allele, but allele which are correlated with the causal and observed causal allele. There may be allelic heterogeneity at the causal loci, or there may be admixture and abnormal and gene interaction factor, like the one we see for the lipoprotein P loci for African Americans for effect on triglyceride. We note also where a similar effect was observed for apolipoprotein for Alzheimer's diseases in Puerto Rican. There are several implications of this and the representation of African. It means that the most genetic risk loci for an allele for blood lipid level are unknown in African descent population. Inter-ethnic differences are still poorly understood. The relative contribution of different factors to this heterogeneity cannot be quantified. And from now, the translational tools of GWAS finding like polygenic score do not work well for African population, and these tests are being deployed in the clinics now. So the objective and approach of our study, our objective is to discover blood lipid variant loci in individuals of African origin, evaluate the performance of multi-thread analysis, because standard GWAS do not take into account the correlation between multiple measurements like blood lipid level. And we want to also explore the replication of the different allele loci between the different African cohorts. Our approach is to meta-analyze lipid GWAS of different cohorts of African descent, perform multivariate analysis, taking into account the relation between the blood lipid thread, and find map the quantitative causal variants. The data sources we collected three main GWAS from summary statistics of lipid GWAS from three main cohorts. The first is the GWAS performed by the African Partnership for Chronic Disease Research, which is a meta-analysis of four African continent-based cohorts from Uganda, South Africa, Kenya, Nigeria, among which we have observational case cohort, but also case control cohort like with Durban case control study and with the African American Epidemiology study. We also have US summary statistics of a million veteran program with African American individual in that study. We also have a summary statistic of lipid measurement from UK Biobank, which are for the Univariate analysis we is performed in two steps. First, we perform a meta-analysis And second, we identify independent loci and try to find map the causal allele. For the first step, we quality control the summary statistic for uniformization across different cohorts, and we apply inverse variance meta-analysis of individual threat, which is a way to weight different studies by the amount of measurement precision of effect size in the studies, and we also perform an heterogeneity analysis. To identify the independent loci, we use 1,000 genome African ancestry sample to estimate linkage disequilibrium between alleles. And the fine mapping is we adopt a Bayesian framework in which we determine 35% credible sets of causality. For a multivariate analysis, we know that in the literature, we have several multivariate analysis which use only summary statistic data and not individual level data. The workflow is following. The summary statistics are combined either with reference panel or individual level information to correlate to estimate the correlation between the different test threads. This is done in order to perform several kind of multivariate tests in which the output may be either p-values of association or a better measurement of effect sizes for marginal threat included in the analysis. We choose to implement the method in the called MTAG, which stands for multi-thread analysis or GWAS analysis. Because it is a summary statistic-based method for joint analysis, it ensures to increase power of association of marginal threat, control for confounding like a population stratification by LD scale regression, and it increase the variance explained by polygenic score. And we choose also it because we can easily compare the result for marginal tests because we have a effect estimate for marginal threat using MTAG, and like other methods for which we only have p-values. The thread of application of MTAG include first estimation of LD score using a reference, an external reference panel. Here we choose with 1,000 genome African subsamples. Second is the quality control of a Univariate meta-analysis before applying it. Application of MTAG, a quality check and identify afterward the independent loci as with the Univariate analysis. And we then compare the multivariate and univariate analysis and annotation by the number of loci, the credible set, and we share through GWAS catalog for novelty, and we make a functional annotation using a website like FIMA or software like AppRank. This is the result of our the harmonization of the GWAS summary statistics we perform. We include different criteria for inclusion of SNPs in a different core. For the APCDR cohort, we just select the SNP which have meta-analysis effect which were estimated. For the Million Veteran Program, we choose SNP which have good imputation quality and which are the same alleles as in the 1,000 genome reference panel. And for the UKB-AFR, we exclude as recommended by the team which perform the analysis to exclude the low confidence SNP which are SNP with minimal allele count lower than 20. And we also include only SNP which have alleles in agreement with 1,000 genome cohort. For 1,000 genome cohort configuration, we select the African subsample and remove first and secondary relative. This left us with 651 samples and 40 million SNPs. And after harmonization, for comparison purposes, we restrict the analysis to only SNP which were present in both in all cohort and across thread, which leave us with about 1 million SNPs because we remove indels too. For the LD score, which is a bit of explanation for what are LD scores. LD scores are the sum of the square of a correlation between a variant and other SNPs in a pre-given windows. It captures the amount of variation which are captured by the focal allele, and it can be used to estimate the heritability of complex diseases from only summary statistics. We observe an increase in the mean LD score from going from 1 centimorgan, which is the window size recommended by the original paper introducing the method to 2 centimorgan. But we observe a plateau to about 20 centimorgan. So we use 20 centimorgan for analysis. This inflation from 1 to 2 centimorgan can indicate a long-range correlation and used by population stratification or admixture in the reference panel we are using. Because the reference panel include the African American, which are an admixed population. So for we perform a meta-analysis using MTAG. For quality check, we look for genomic control, and we intercept for the LD score regression, which quantify the amount of inflation of summary statistics, which can be attributed either to polygenicity or stratification. This ratio gives an account of the amount of inflation of test statistics, which can be attributed to polygenicity and not stratification. Yes, usually ratios near zero are preferred to indicate that there are minimal amount of stratification. Here we see that the ratio for these two sets are a bit elevated, but the intercept is not significantly different from zero. So we conclude that there is not much stratification remaining in the summary statistic after we perform a meta-analysis using metal. These are the results for instance, for the meta-analysis for HDL. See that in total we observe 31 genetic loci, independent loci, in which we had 162 independent lead loci. These the Manhattan plot indicate that these leads are on chromosome 1, 2, 1, 2, 3, 4, 17, 18, 16, 14, and 13. And arrangement analysis indicate that the SNPs were significant, and the gene, because the gene associated with them tend to be overexpressed in the liver, which is expected given the implication of the liver in lipid metabolism. So, in addition to canonical loci of blood lipid thread, we find several non-reported loci. For instance, this is an example. We find a loci on chromosome 17, which is polymorphic in Africa, but not in other continents. And this was located in the MPP2 gene, which is associated with in GWAS catalog with bone mineral density, and bone mineral density have been linked between to blood lipid level in previous studies. There are several loci like with this profile in our meta-analysis. In the comparison, we see that in general, multivariate analysis boosts the marginal association for marginal association. We see that when we compare MTAG, LDL effect sizes and p-values for MTAG compared to univariate analysis, we see that we detect much more loci for the MTAG approach than the univariate approach, which was expected. And also we observe after performing meta-analysis, we select for each trait the lead SNPs, the independent lead markers, and test the correlation of effect size between different cores. Between we have APCDR and the million veteran core, APCDR and UK Biobank core, and the million veteran program and the UK Biobank core. We see that in general, there is a good agreement in the effect sizes using the formula we used, which corrects for winner pools and estimation noise, because we have different sample sizes for the different core. We see that in general, correlation tends to be higher between between the UK Biobank and Million Veteran Program, despite a lower sample size of UK Biobank compared to APCDR, which this can be due either to greater genetic similarity between participants in Million Veteran Program and UK Biobank, because these are both composed in the majority of admixed individuals, admixed African European individuals, and APCDR is a heterogeneous score from the continent, which includes also this control study, which can also explain the difference in correlation. But the correlation is high, which means that we are measuring the same effect across the different cores for the SNP which were significant in the meta-analysis. Up next, we want to finalize the annotation of and perform a fine mapping. For as a sensitivity check, we will run the same analysis within each core separately and meta-analyze compare with this analysis. We also want to relax the criteria for the inclusion of SNP in the meta-analysis, because we remove indels, and these indels are maybe the causal alleles. This can bias our fine mapping approach. So we will relax the meta-analysis, and also this will allow us to make a much more in-depth comparison between the different cores. We'll also perform a sensitivity analysis to LD score regression for the estimation of a correlation matrix used in MTAG, because as we say that we remark a marked inflation of LD scores from going from 1 centimorgan windows to 2 centimorgan windows, and like in the original paper for the LD score in European reference panels. In summary, as we said, there is a lack of transferability of lipid loci in African ancestry. We perform a meta-analysis of lipid measurement in 3 cores. We replicate the canonical loci of lipid and uncover new candidate loci. We determine the correlation of effects at the cross cohort and note that they were high. We confirm the higher power of a joint analysis. I would like to finish by thanking Dr. Shadun Fatumo, my supervisor, and Dr. Shegnas, my assistant advisor, Dr. Jeffrey Schaefer, who is my mentor at Touré University, along with Dr. Jian Li, Dr. Tina Shikore for his comment and advice, Professor Wele of the director for our center, a team of the Biostatistics Department at Touré, and the HPC team in which I perform the analysis. And I would like to thank everybody at the African Center in Excellence in Bioinformatics. Thank you for your attention.
Thank you so much, Sounkou. All right, so I'll ask, the floor is open for questions directed both at Martha and at Sounkou. I see from the chat box, there's already some questions for Martha. So Rita has said, nice work, Martha. How many target amplicons did you multiplex for?
Yes, so we targeted multiplexing was based on the primer annealing conditions, because the different primers had different annealing temperatures. So we picked the first thing that I perhaps didn't mention is that we could not put subsequent primers in the same pool because they had a region that overlapped. So putting them in the same reaction would preferentially amplify the shorter region as opposed to the larger region. So we picked non-subsequent applicants that had similar annealing conditions. And for using that criteria, we had either two primer pairs or three primer pairs going into a single well. And so we had either three or four reactions per sample, as opposed to now 8 reactions per sample.
All right, thanks, Martha. And then another question for you is from Kahinde, who says, Martha, good one. I think I missed the earlier part of your talk. I just want to know whether the phylogeography was discrete or continuous.
That was discrete phylogeography.
All right, thanks. So are there any other questions also directed at Sounkou from the audience? You are also welcome to unmute yourself and raise your hand and ask your question. I have a question for Sounkou. Sounkou, I think in the last few of your slides, you mentioned that you can relax the criteria for the meta-analysis. Is that standardly done? And then what is the standard that they use for the criteria? And then how do you choose by which measures to relax it with?
Yes, I will, because for the analysis, in order to compare the univariate and multivariate analysis, I remove indels and restrict the analysis to alleles for which the difference in frequencies are not are lower than 15% between the cohort, which will limit us to only alleles which are either common or very similar in frequency between the different cohorts.
Okay. All right, thanks. We still have some more time for questions. Anybody?
Yeah, hello, Rolanda.
Yes, thanks, Albert.
Yeah, I have a question for Martha.
Go ahead.
Okay. Yeah, so I saw that you talked about VP42. I want to know if it's unique to HRV.
Okay, thank you for the question, Albert. So the VP42 region, yes, it's a region that's about 400 bases long, and it's actually what is used to assign genotypes. So this means that for every genotype, the VP42 is characteristically unique, and so is the VP1. So I mentioned that studies either use the VP42 or the VP1. Both regions show congruence when it comes to genotyping, and so this means that the fact that they're used for genotyping, it means that they are unique to every genotype.
Okay, thank you very much.
I hope I answered your question.
Yes, yeah, you did.
Okay.
Thanks, Martha and Albert. Are there any other questions? Audience members are very quiet. All right then, so if there's no more questions, I just want to thank the speakers for their presentations today. It was really interesting. And I also want to thank the audience members who joined the call today. Thank you so much for your attention. I'm not sure if there's some announcements from Verena and Albert, who also helped organize this session.
Yeah, I don't have any from my end.
All right, thanks, Albert. And Verena, do you have any announcements before we call it a day? Not sure if Verena can hear me. But all right then, thank you so much, everybody. It's really nice to see all the progress made from the fellows. And I'll speak to you in 2 weeks' time for the next seminar. Right, have a good day further. Bye.
Bye.
Bye-bye.
Rina, you can close the meeting.